Hello everyone, Assalamu alaikum. Welcome to the new lecture on pharmaceutical chemistry. In this video lecture, I am going to discuss with you about the electrochemical methods. This is part of pharmaceutical analysis. Electrochemical methods. Electrochemical methods are those analytical methods in which we are going to analyze or determine the electrical properties of our sample or analyte. So, by analyzing the different electrical parameters of the sample like charge, electric potential, current that it carries or the EMF generated during the reaction, we can know or determine the exact concentration or amount of the analyte that is being consumed or produced during the particular type of chemical reaction. So in this way we can also determine the electrode potential of our cell, that is the electrochemical cell, and the electrode potential is basically also useful in determination of the analyte or sample concentration that is being formed at the electrode surface, that is basically the reduced or oxidized form of our sample. So in this way, the sample or analyte that is being used in the electrochemical cell undergo through a chemical reaction, that is usually the redox chemical reaction. So after that, the concentration of analyte will vary in the electrochemical cell. It will be different at the surface of electrode, and it will also be different in the bulk of electrochemical solution. Current is usually, you know, that it is basically the measurement or the flow of electrical charges. So we can also say that it is basically measure of the rate of analyte oxidation or reduction, means when the sample or analyte undergo through the chemical reaction during the electrochemical cell, that is usually the oxidation reduction reaction. So then it can cause the current to flow inside the electrochemical cell. Electrochemistry. Electrochemistry is that branch of chemistry that is deal with the different type of chemical reactions that are used to generate the electricity. Or it is also associated with those methods which use the electric energy, and in return they are going to cause the certain type of redox reaction in the sample compounds. So basically electrochemistry is the study of all those chemical processes that cause the flow of electron or charges, or that cause the measurement of the different type of electrical properties. In flow of charges, we know that it is the electricity, and for the current to measure there must be a particular type of drift velocity, because there are the two fields of electricity, like one is the electrostatic, other is the electrodynamics or electricity. For electrostatics, our electrons or charges flow at the uniform or constant velocity, but for current to produce, it must have a drift velocity, means that is a non-uniform velocity which produce the drift acceleration in the charges or electron. Then in that case current can be generated. So in this way, due to the movement of the charges, the redox reaction is produced in the electrochemical cell. Electrochemical methods. There are the main two type of electrochemical methods. So broadly, electrochemical or electroanalytical method can be divided into the two type. One is the interface method, other are the solution methods. Interfacial methods are those electrochemical methods which responds directly or indirectly to the presence or absence of analyte on the surface of our electrode, means they are going to deal with the surface phenomena. And which surface? That is the surface of our electrode. Electrode, we know that is part of the electrochemical cell, means two different type of electrode, one will be the anode, other will be the cathode. So during a chemical reaction, if on one side oxidation will occur and on other side reduction will occur, so during that reaction the amount or concentration of analyte that is going to be present on the surface if they are going to be studied, and that phenomena is known as the interfacial methods. Interfacial methods can be divided into the two other subtypes. One are the static method, other are the dynamic method. As the name is indicating, static methods are those means which do not have any disturbance or in which the current is zero. I is used for representing the electric current. Means static interfacial electrochemical methods are those methods in which there is no any flow of current. Clear? So the example of static electrochemical method is the potentiometry. Second interfacial method is the dynamic method. Dynamic means it is indicating that what? That something is flowing. And in sense of electrochemical method, the charges are flowing, and we know that flow of charges is the current. So in this way, dynamic are those method in which the surface of electrode or the that is the inductor, electrode receiver, and electric stimuli with a non-zero current. Means in them the current is flowing. The dynamic electrochemical methods are of two different types. Among them, one is the amperometry, voltametry, calorimetry, and the control potential technique and the titration methods. So basically what we have learned that interfacial electrochemical method are of two types. One is the static, in which there is no any flow of current. The example is the potentiometry. So means in these interfacial, static, and potentiometry method, we determine the electrical properties of our sample without any flow of electric current. But in dynamic electrical methods, we will study what? The flow of current. Means we will determine the electric property by the flow of current. Due to the flow of current, the electrical properties can be studied. And their example is voltammetry, amperometry, and so on. So this table is representing the different type of electroanalytical methods and their properties which they are being used to determine for the analyte. Like this column is representing the type of the electrochemical method. Over here you may observe these are the different type of electrical properties that are being measured with the help of these methods. For example, for number one is the potentiometry. Potentiometry is that electroanalytical method which is used to measure the potential difference of our analyte. So potential difference, we know it is being measured in the units that are the volt, where V is for the volt. So this column is representing the measurement unit of that particular electric property. Second method is the conductometry method. Conductometry methods are those which are used to determine the ability of analyte to whether the conduct or resist the current or flow of the charges. So conductometry method are being used to measure the conductance or resistance of our analyte. Conductance is ability to cause the flow of electric current, and resistance is what? Resistance is the opposition, means to resist the flow of current. So the unit for resistance is ohm. Amperometry and voltammetry are those electric methods in which we measure the electric current as a function of applied potential, and for that we use the electric current is being represented by the I letter. Coulometry is used to measure current as a function of coulombs, that are the charges. So coulombs charges, for that C is equal to I is the unit. Capacitance is the ability to measure, capacitance is the ability of any conductor to store the electric charges, and we know that the unit of capacitance is the farads. So F is equal to C into V inverse. So these are the different type of electroanalytical method and their application in the pharmaceutical analysis with respect to our sample analyte. So basically as far as the brief history of electroanalytical methods are concerned, during the 1889, Nernst proposed the equation which states that the electrochemical cell potential is directly proportional to the concentration of solution. So he proposed equation on the basis of which he said that electrochemical potential of any cell, or what we say the electrical properties of any analyte, is depending on its concentration that is present in the solution form in the cell. Later on in 1906, another scientist named as Kramer explained that the potential difference that is existing between the two different electrodes is responsible to cause the electrical properties of the analyte. And the two different electrodes are, one is the reference electrode, other is the indicator electrode. Reference electrode is that electrode whose potential is known or constant. Indicator electrode is that whose potential varies according to the sample or analyte. In 1909, another type of electrode was discovered, that is the pH electrode. That is the type of the glass membrane electrode, and by this the application of the pH glass electrode is the measurement of potential of the solution. So now let's discuss here briefly the principle of the different electrochemical methods. Like number one is the potentiometry. Potentiometry, I have already told you that it is one of the static interfacial methods in which we are going to determine the potential of our sample but without the flow of current. So there are the two types of the potentiometry. One is the direct potentiometry, second is the indirect potentiometry. Direct potentiometry is that in which we determine the potential of our analyte by using the Nernst equation. Means during the direct potentiometric method of analysis, we conclude that we can conclude the result as the concentration of analyte is directly proportional to the electrode potential of the cell. And on the other hand, indirect potentiometry is used for determination of the change in potential, which is usually the end point. That is basically one of the methods of the titration. So the difference between them, in direct potentiometry, our potential is dependent on the concentration of analyte, whereas the change in potential in indirect potentiometry is based on the endpoint result that are basically helpful in the titration method. The second method is the colorimetry. The principle of this method is it determines the quantity of our electric current or the electricity. Voltametry is also used for determination of the current. This is totally opposite of the potentiometric method because this involve the this involve the flow of current. The only difference is by altering the potential, because electric current can be generated once the potential is being varied. If potential flow with the constant flow, then no any current can be generated. This will cause the change in current or the flow of current inside our electrochemical cell. Conductometry is basically used for the measurement of the conductance of our electrochemical cell. Redox reaction. So now let's discuss what is the redox reaction, how it takes place inside our electrochemical cell. A redox chemical reaction is basically that chemical reaction that causes the change in oxidation state of one or more element, or what we say here, that reaction that causes change in oxidation state of our sample or analyte. Oxidation state, we know that the oxidation number is the total number of electrons that are either being gained or lost during the chemical bond formation or breaking. So we know that when the two chemical species react with each other, then one of them is going to lose the electron, other is going to gain the electron. So during this chemical change, their oxidation number will be changed. Oxidation number of one species is going to be increased, and for other it is going to be decreased. So the substance or the chemical species which loses an electron, its oxidation state will increase, because we know that oxidation is that process in which electron is being lost by the chemical species, and that chemical species that gains the electron, its oxidation number will decrease, because it is going through the process of reduction, because it is going to be reduced. And the entire chemical reaction combinedly is known as the redox reaction, means the reduction and oxidation reaction. So redox reaction. So redox reaction is basically caused by the two chemical species. One of them that is going to be oxidized, other is that is going to be reduced. So in the language of chemical analysis, or what we say the analytical chemistry, there are the two different terminologies used for these chemical species. One is the reducing agent, other is the oxidizing agent. The species that is going to be oxidized is known as the reducing agent or reductant. Means the species whose oxidation state is going to be enhanced, whose oxidation number is increased, which is themselves are going to be oxidized, are known as the reducing agent or reductant. Means they are causing the reduction of other chemical species. Clear? So over here you may observe this is the A is the reducing agent, because it is the losing electron. They are causing the so compound A loses the electron. So in this way they are going to be oxidized. Number two is the oxidizing agent or the oxidant, are those chemical species that are going to be reduced. Clear? So they themselves are going to be reduced, they are going through the reduction process, but they are causing the oxidation or oxidation of the other chemical species that is involved during the chemical reaction. So in this chemical reaction you may observe that this B is the oxidizing agent, because it is causing the reduction, it is going to be reduced by itself and causing the oxidation of other chemical species. So B themselves are going to be reduced because they are gaining the electron. So in this case, in the previous example, the reaction shown, hydrogen is being oxidized, so we can say that it is the reducing agent, because it is causing the reduction of other chemical species, that is the fluorine. And fluorine is said to be reduced, and we can also say that it is the oxidizing agent. So this is again over here the difference between the reducing agent and oxidizing agent. A reducing agent is that which themselves is going to be oxidized, means it is losing some of its electron, and oxidizing agent is its opposite, which is gaining the electron, means they themselves are going to be reduced but causing the oxidation of other chemical species. So during the oxidation state of reducing agent is going to be increased, and for oxidation state of the oxidizing agent is going to be decreased, because they themselves are undergoing through the reduction, and whenever a chemical species undergo through the reduction, its oxidation number will be decreased. Reducing agent are the electron donor, because they are going through the oxidation, they are giving the electron to the other chemical species, and the oxidizing agent are the electron receiver, they are gaining the electron. Oxidizing agent the reducing agent are themselves going to be oxidized, and the oxidizing agent themselves reduced. And other point is the reducing agent cause the reduction of other reactant, or what we say other chemical species, and again the same point, the oxidizing agent cause the oxidation of other chemical species. So now let's discuss about the electrochemical cell. So we have learned the basics of the electrochemistry, different terminologies, redox chemical reaction, that what is oxidation, reduction reaction, what are the different type of electroanalytical method and their basic principles. So now let's learn about the what is the instrument that is used for those different electroanalytical methods. So the instrument that is used is known as the electrochemical cell. Electrochemical cell is basically that device that is capable of either generating the electric current from the chemical reaction or that uses the electric energy from external source to cause the chemical reaction. So their purpose is to either generate the electric current or to use the electric current in order to cause the chemical change in the simple analyte solution. So that chemical reaction will be the redox chemical reaction. So the example, let's take as the simplest example, that is the flash battery cell can act as the electrochemical cell. So it is something that is going to change the chemical energy into electrical energy or electric energy into chemical energy depending on the type of electrochemical cell. So basically it consists of the two compartments. These two compartments consist of the electrode and they are going to be submerged in the solution of the electrolyte. Electrolyte is our analyte or sample. Electrode that is going to be used in an electrochemical cell is the conductor, means which must have the ability to conduct the electric current. So usually it is made from the metal that can conduct the electric current, and the electrolyte is usually the fluid that have the ability to conduct the electricity. The chemical reaction that is going to occur inside the electrochemical cell is known as the redox chemical reaction or the oxidation reduction reaction. So the electrochemical cell, what we have learned that it is any type of instrument that is consisting of the two different type of electrode which causes the flow of either the current or would generate the current due to the electrochemical reaction, that is the redox chemical reaction, oxidation reduction reaction. So now let's check out what are the different type of electrode that are present. Usually the two electrode are present, one has the positive charge, other has the negative. So these are known as the cathode and anode, and these two electrodes are connected with each other by the help of wire, and in this way electrochemical circuit is completed. So usually a metal strip in the battery, that is the copper, is act as the cathode, and which is the electrode where the reduction take place. So if we are taking the battery as an example of our electrochemical cell, so in that the copper is work as the cathode electrode, and cathode is that electrode where reduction or gain of electron is going to take place. And the other electrode in that is the zinc electrode, which acts as the anode. Anode is that where the oxidation is going to take place, means electron are going to be released from that chemical species. Types of electrochemical cell. There are the two main type of electrochemical cell on the basis of their principle. One is the electrolytic cell, second is the galvanic or voltaic cell. Electrolytic, let's break this terminology, that electro means the electric current, it is representing or relating with the electric phenomena, and lytic means its lysis or breaking. So electrolytic is that electrochemical cell or device which uses the current from the external source or electric energy from external source. And why this energy is used? Because it is going to cause the electrochemical or redox chemical reaction inside our electrochemical cell, and by that the potential of the sample or the potential can be measured. So basically electrochemical cell in which the non-spontaneous reaction is driven by external source or current is known as the electrolytic cell, and it convert basically electric energy into chemical energy. The chemical energy that is going to be generated is basically the redox chemical reaction that is occurring inside this chemical cell. The second electrochemical cell is the galvanic or voltaic cell. This is totally opposite of this electrolytic cell. Galvanic cell is basically that electrochemical cell which convert the chemical energy that is produced during the redox chemical reaction into the electric energy. So there are the different names that are used for the galvanic cell. So they like it can be also called the Voltaic cell because of the discovery by the two different scientists, that is Luigi Galvanic and the Alessandro Volta. The different example for the galvanic cell are the batteries or the fuel cells. So now let's check out the difference between the galvanic and electrolytic cell. So we have already discussed their principle that electrolytic is consuming or using the energy, electric energy and converting into the chemical energy, and galvanic is using the chemical energy and converting into the electric energy. So the other difference is in galvanic cell the anode is negative and cathode is positive, and in electrolytic cell anode is positive and cathode is negative. The reaction that is going to occur in galvanic cell is spontaneous reaction because in electrolytic cell the non-spontaneous reaction is occurring because in this electro current is being supplied from the external source, and that is going to be converted into the chemical energy. And the other difference is galvanic cell does not require the external voltage potential. On the other hand, the electrolytic cell requires the external voltage potential. So this is again the diagram for these two cells. In this you may observe this is the galvanic cell, this is one of the negative electrode, this is the positive electrode, and this is anode, this is positive, because we have learned that anode is negative in the galvanic cell and cathode is positive. And this is the electrolytic cell of electrolytic cell diagram. You may observe the anode has the positive charge and the cathode has the negative charge. So it uses the electric energy and convert into the non-spontaneous redox reaction. Whereas in the galvanic cell, the redox chemical reaction that is occurring spontaneously inside the electrochemical cell is going to be converted into the electric energy. Voltaic cell. Now let's discuss the what is the Voltaic cell, what is its principle, what are the different parts of the Voltaic or galvanic cell. A Voltaic cell is basically discovered by the galvanic scientist, and it is basically that electrochemical cell that uses the spontaneous redox chemical reaction and convert it into the electric current. So basically there are the different parts of this Voltaic cell. Most important are the consist of the two separate half cell. Each half cell has an electrode. Electrode, we know that it is basically a strip of metal, and electrode is immersed inside the electrolyte solution, that is the solution of our sample. So these two half cells are linked together by the help of wire from electrode 1, which is basically going from one electrode to the other electrode, and a salt bridge is used, which is usually used to connect both of these half cells. So let's represent that how can we represent the equation of electrochemical cell. Anode is usually considered as a negative electrode, and it is that electrode which is present on the left side of our voltaic or galvanic electrochemical cell device, and cathode is positive and it is mostly present on the right hand side. So in this diagram you may observe this one is the zinc electrode, this is the on the left hand side, and it is acting as the anode, and it is that electrode on which oxidation is occurring. And this is on the right side is the copper electrode, which is acting as a cathode in case of the galvanic cell. The salt bridge is this bridge which is going to be connect the two half cell. This is the one cell, this is the second cell. These two separately are known as the half cell. So these two half cells you may observe are going to be connected by this electric wire and also by the help of this salt bridge. Salt bridge maintains the neutral composition of both of these electrolytic solution. And if we represent the Salt bridge in the equation, we use the two parallel lines. So the components of Voltaic cell are the half cells. Half cell is the which consists of the two separate half, each containing a different electrode. One is the anode, other is the cathode. So on both of these half cell, two different type of chemical reaction are occurring. On one side oxidation, other side is the reduction. So basically each half cell is converting what physical or chemical energy or physical or chemical process into the electrical energy. So on one side oxidation is occurring, on other side reduction is occurring. So entire cell reaction will be the redox reaction, and each half cell reaction will be either oxidation or reduction. Electrodes. Electrode is any strip of metal on which the reaction is going to take place. Which reaction? Redox chemical reaction. In a Voltaic cell, oxidation and reduction occur on the surface of the metal. So like on the cathode is that electrode where reduction is occurring, and on anode oxidation is occurring. So electrodes are basically a strip of metal, and they are going to be immersed in the electrolytic solution, and that solution have the capability to either conduct the electric current. And the electrolytes are usually the ions in the solution that they are usually contained in the fluid form, and it has our sample. And there are the two possibilities that can occur in the electrolyte solution, either the oxidation reaction or the reduction reaction, depending on the type of electrode. Like the anode is that on which the oxidation reaction take place. Like in case of Voltaic cell, the zinc electrode is the oxidation, it loses the electron, and cathode is the copper electrode which gains the electron. So in this way we may observe the flow of electron will be always from the anode to cathode, means from left hand to the right hand of the cell, because the zinc is going to lose the electron and it is the electrode on the left hand side. We may also say that the electron will flow from more negative half reaction to the more positive half reaction. More negative half reaction is the anode side of the Voltaic cell, and the more positive is the cathode side of the Voltaic cell. Salt bridge. Salt bridge is the most important component of any of the Voltaic cell. It is basically composed of the electrolytic solution of potassium nitrate or potassium chloride, that is basically the salt of potassium. Clear? Why we are using the salt bridge? Salt bridge is used to connect the two different half cell, and its purpose is to keep the solution in the neutral form, and it allows the free flow of ion from one cell to other cell without changing their neutrality. Without the salt bridge, the positive and negative charges will build up around the electrode and the reaction will be stopped. So means we can also say if salt bridge is not being used, then our Voltaic cell cannot perform its function, because now the positive and negative charge can build up entirely around both of the electrode, and no any type of oxidation reduction reaction will take place. So basically it prevents the transference or diffusion of solution from one cell to other cell. It connects the two half cells and completes the circuit and prevents the liquid-liquid junction potential, that is the potential difference that arises between the solution when contact with each other. This point I have already explained over here, that if we are not using the salt bridge, then the positive and negative charge will build up around both of these electrodes and no any type of redox reaction will take place. So now let's discuss the principle of Voltaic cell. Voltaic cell is consisting of the two half cells that are joined or connected by the electric wire and also by the help of the salt bridge. So the two different electrode on each side of half cell, one has the anode, that is the zinc electrode, and other side, that is on the right side, has the cathode electrode, that is the copper electrode. The electron will flow through the external circuit or wire, and the electron will always flow from the zinc to the copper, because zinc is going to be oxidized, zinc is going to lose its mass, and copper is going to gain its mass. And because on the copper electrode on the right hand side, reduction is going to take place. So in this way, the concentration of zinc sulfate will increases, and the concentration of copper sulfate is going to be decreased. So in the solution in both compartment is in neutral, will be in neutral form because of the presence of the salt bridge. And during the passage of electric current, the electron will always flow from the zinc electrode to the copper electrode. So in this way, we may measure the potential of the cell on the basis of the flow of electric current. So this is the again diagram for your understanding, that this is one for suppose the diagram for the Voltaic cell. In this you may observe that this one is the zinc electrode, that is the anode. You may observe that this zinc is undergoing oxidation, it is losing electron, and this electron is flowing through this external wire and is coming in the copper, and copper is gaining this electron, and that's why copper is undergoing through the reduction. So this entire, these are the two half cell. This is one half cell on which the oxidation is occurring, on the second half cell reduction is occurring. So this is entire Voltaic chemical cell is causing the redox chemical reaction to convert into the electric current, and due to the flow of electric current, this bulb is glowing. So we may represent the equation for the Voltaic cell or galvanic cell in the following way according to the reaction. So zinc, as it is going to be continuously oxidized, so zinc is continuously oxidized, it is losing electron, and in this way it has the discharge. And on the other hand, the cathode half cell reaction can be represented like this, that copper is going to be reduced, it is gaining the electron, and copper to electron are being gained. First copper has the positive charge, and it is gaining electron, this charge will be neutralized. Electrode potential. Electrode potential, this is basically the terminology for your understanding. This is not the part of the principle of the Voltaic cell. Clear? So electrode potential is basically the potential difference that is set up or generated between the two metal strips, or what we say the two different electrode. And it is basically the tendency of an electrode to lose or the gain electron, means the tendency or capacity of any electrode to undergo through the oxidation or reduction when it comes in contact with the solution of ion, or when it comes in contact with our electrolyte solution. So it can only be measured by using the sum of the electrode as reference. So now question arises that electrode potential is basically the type of potential that is generated inside the chemical cell due to the oxidation or reduction reaction. But how can it be measured? So we may measure the electrode potential of electrochemical cell once we have one of the electrode which is acting as the reference electrode. So in that comparison, we may measure the electrode potential of other cell, because reference electrode is that electrode whose potential is already known, whose potential is constant. The reference electrode types are the different type of examples of reference electrode are the standard hydrogen electrode, are the saturated calomer electrode. These are the different type of the reference electrodes. Electrodes used in the electroanalytical methods. So there are mainly three types of electrode that are used in the electroanalytical method. One is the working electrode, second is the reference electrode, third is the auxiliary electrode. Working electrode is also known as the indicator electrode, because it is that type of electrode which is going to measure the potential of the sample, or means it is going to indicate the change in sample concentration, means its potential will change according to the change in the sample concentration. So on working electrode, our reaction of interest is occurring. So means working electrode is that electrode on which either oxidation or reduction reaction will take place. Clear? So with respect to that redox reaction, its potential will vary, And why it is going to be vary? Because the sample concentration is going to be changed. The best example of working or indicator electrode is the glass membrane electrode, that is the pH electrode. Reference electrode is that electrode which produces a constant potential, means its potential is already known. Means if we are immersing our sample solution or any other electrolyte solution, its electrode potential is not going to be affected. It will remain the same constant. The example of reference electrode is saturated polymer electrode and silver chloride electrode. Third electrode is the auxiliary electrode. This electrode is used as a counter electrode, and it is used in a 3 electrode system, and it is mostly used in the voltammetric determination. The example of auxiliary electrode is the standard hydrogen electrode. So when we are using the 3 electrode system, in that case saturated standard hydrogen electrode will act as the auxiliary electrode, and that is the counter electrode, and the 3 electrode system is usually used in the voltammetric determination, and the 2 electrode system like the working electrode and reference electrode are mostly used in the potentiometry measurement. Standard electrode potential. Electrode potential, we already observed that it is the capability of electrode to undergo through the oxidation or reduction to whether gain or loss the electron. So the standard electrode potential is basically electrode potential which is measured under the standard condition. Which type of condition? Means the solution of our sample must be 1 molar concentration, and the temperature for that is the 298 Kelvin. That is the, in sense of, if we are considering the degree centigrade, will be I think 25 degrees centigrade. Clear? And the pressure for the standard electrode potential must be the 1 atmospheric pressure. So whenever the electrode potential of any cell is measured under these 3 conditions, then it will be known as the standard electrode potential. Expression of Nonstitution. So now let's learn that what is basically the Nonstitution, why it is being used. Nonstitution is basically the equation which is showing the relationship between the capacity of an atom or ion that is going to either take up electron, means it is used to measure its reduction potential, that can be measured at any of the condition. Means any condition means other than a standard condition are 1 molar solution, clear, 1 atmospheric pressure and 25 degrees Celsius. But through Nernst equation we can measure the potential at any non-standard condition. So means our sample solution can be any and the temperature can be other. So this is the formula for Nernst equation, that E cell is equal to E naught minus RT in Q. So Nernst equation provides basically the relationship between the cell potential and the of electrochemical cell and the standard cell potential. The C naught is the standard cell potential, temperature and the reaction quotient. So by this equation we can measure the cell potential of any electrochemical cell under the non-standard condition. So Nernst equation is also useful to calculate the electric potential under any of the non-standard condition. Means under any solution, it means our concentration of solution can be any other than 1 molar, temperature can be other than 25 degrees Celsius, and pressure will also be different. This equation was given by the German chemist, that was the Walter Hermann Nernst. So this is the formula for Nernst equation, that is the E cell is equal to E naught minus RT in Q. So in this, E cell is basically the potential of electrochemical cell. E naught is the cell potential under the standard condition. R is the universal gas constant, T is the temperature, N is the number of electrons that are being transferred in the redox chemical reaction. Clear? And the Q is the reaction quotient. So let's check out the what we are using the Nernst equation. Nernst equation is very important in the electroanalytical methods. There are the different applications. By the help of Nernst equation we can use to calculate the reduction or oxidation potential under any condition. Clear? So, and we can also calculate the standard electrode potential We can also compare the relative ability of the different chemical species to whether it will undergo oxidation or reaction. And we can also identify a single electrode that can produce the electric potential. We can also measure the EMF of an electrochemical cell. We can identify, determine unknown ionic concentration. And beside this, we can also measure the pH of solution. Solubility of sparingly soluble salts can also be measured through the application of Nonstitution, but for that we have to use the electrochemical cell. Now let's have a quiz. So this is the quiz for your self-assessment. You have to attempt the quiz in the comment section. Question number 1, define electrochemical method. Question number 2, differentiate between electrolytic and galvanic cell. Number 3, name the anode and cathode electrode of galvanic cell or Voltaic cell. Number 4, what is standard electrode potential? Number 5, define indicator electrode.